just want to say first here that this what I'm about to present to you is a result of a partnership between a university, so the Ross University School of Veterinary Medicine here in St. Kitts, the Caribbean Initiative that jointly funded this project, and it's basically an NGO which funds Caribbean students to do research in the Caribbean, and the Department of Marine Resources, and they were responsible for granting the permissions and so on for this project. So we're just going to go straight into it. Now, there are few pathogens and parasites reported in the Caribbean spiny lobster. I have a few, for example, Cymatocarpus soliaris was found in Mexico. Panulirus argus virus 1, I believe most of you should be aware, or those of you more to the west would be aware of Panulirus argus virus 1 is found as far east as Puerto Rico. And we also have microsporidians. So this is just basically what's known in the literature. And as we know, this is a very lucrative fishery right across the region. So this lobster is found as far north as the Carolinas and as far south as Brazil, and it generates about almost $1 billion US in revenue throughout the region. So the two main aims of this project were to conduct a health screen on the St. Kitts population, essentially doing disease surveillance. We need to know how healthy our stocks are, and also to examine reproduction in the species and to determine the size at sexual maturity and peak periods of reproduction. I spoke earlier about closed seasons. At the moment, St. Kitts does not have a closed season. However, they restrict fishery. There is a minimum size that you can take the lobsters at, which is 95 millimeters carapace length. And this has been, this is enforced here in St. Kitts and in the Eastern Caribbean. I believe other countries within the region have slightly different sizes, but that is the size that St. Kitts and the Eastern Caribbean has. So we're going to go into the lab. So we've collected our lobsters from the fishermen. We're in partnership with the fishermen, and we have done our dissections. We've done dissections. We're looking at any abnormalities that the lobsters might have. So these are we're doing several kinds of observations and analyses. So we're looking grossly. So we're just looking with our naked eye to see whether there are any abnormalities. And then we're looking under the microscope to see at a cellular level whether there are abnormalities. And if we see anything, we look at the DNA. So we try, for example, if we see something that might indicate some sort of disease, we do PCR to amplify the DNA to see whether we can identify what that parasite might be. And we collected muscle samples, gonad, gills, and hemolymph. For the sexual maturity study, we looked at the ovaries grossly to see what level of sexual maturity the ovary is at, what level of maturity the ovary is at, and also on a cellular level, physiologically, what stage of development the ovary is at as well. So we're just going to move on and just bring that together. I have also to say that we looked at the hemolymph, and you'll see in what section we looked, we did our analysis on the hemolymph of the lobsters. So some quick results, just an overview. We found a worm. So the photo in the middle, there are some, if you look within the blue shape, the oval, you would see these little worms within the eggs of the lobsters. So these are called nemertine worms. They eat the lobster eggs by puncturing the egg and sucking the contents of the egg. So on the right, the structure within that circle is what they call the stylet, and that's what the worms use to puncture the eggs. And if we have many worms or worms eating the eggs, that means that we would have less lobsters possibly coming to maturity. being able to grow, go through the life cycle and become adults. And this kind of worm has been responsible for the collapse of fisheries, crab fisheries in Alaska and California. We have also a microsporidium called Amosine hernkindii. It's a new species. If you look to the left, you would see the muscle of the lobster looks white. And if you cook a lobster, the muscle turns white. So basically, this lobster looks as if it's been cooked, but it actually isn't cooked. And that is what we saw grossly. And under the microscope, we saw unicellular parasites within the muscle cells. Now, this is a novel species described in Florida and also in St. Kitts. And based on that, it is quite possible that there is a wide geographical range. So this parasite might be found not just in these two countries, but also the countries in between and probably even beyond that. And it is either rare or underreported. We found this parasite in 2 out of 313 lobsters that we analyzed. I want to go back really quickly and also say in the case of the nemertine worm, this was reported in Florida in 2017, in Colombia in 2018, and we also found it here in St. Kitts, which also points to possible wide geographical range. I mentioned before Panulirus argus virus 1. It is the only viral disease reported in Panulirus argus, and it is lethal to juveniles. You would find then that the juveniles would die more easily than adults would. And this has been found first in Florida, first in Florida, and it kind of spread over most of the Western Atlantic area over time. And it is, if you look at the picture on the right, the hemolymph itself turns white. That's how you can tell that the virus is there. The juveniles also become more lethargic and they eventually die. However, we did not find that in St. Kitts, thankfully. Moving on to sexual maturity, we looked at the ovary, as I said, histologically and grossly, and we were able to stage the sexual maturity. And what we also did was we tried to determine the size that's physiological and functional maturity, which is the size at which 50% of the lobsters become mature. And we also identified that the peak maturity occurs between March and May. And we believe that each territory should conduct this sort of research, and it should be current. So I know that there are some countries that have done this sort of thing, but it was done like 5 years ago, decades ago, but the climate is changing, fishery practices are changing, so fishing pressure would change as well. So we're suggesting that be the case. Belize did something similar. What they found out is that their minimum legal size was actually what was it? It was actually smaller than their sexual maturity, which means that the lobsters would not be able to reproduce at least once before they're taken out of the fishing stock. So future work, it would be good to expand this research and be able to tell what other countries within this region have the nemertine worm, or also may have the microsporidium that we were talking about. It would be also good to identify all the species of nemertine worm that might be here. Disease surveillance should be ongoing because we want to make sure that our stock is healthy so we can have sustainable fisheries. And it's also good to partner with fishermen and encourage them to report the abnormalities. And when we get those reports, we can collect the samples and do analyses and try to figure out what may be responsible for the diseases or the changes in behavior, changes that we might see in the fishing stock. So that's it. If you have any questions, feel free.
Thank you very much, Ms. Herbert, for a very informative presentation. It reminds us that many things need to be considered when it comes on to managing our stock sustainably, including diseases. and the variations in these diseases from year to year over time. So it is something that we truly need to monitor